the southern resident populations down here. The southern residents are the most southerly distributed of this group. And they're also somewhat anomalous in that they're one of the only groups that is sort of not recovered well after being protected by the Marine Mammal Protection Act in the mid-1970s. So for the most part, killer whale populations are actually doing pretty well and have been increasing. There are a couple of exceptions, the southern residents being one of them. So focusing on even further, so we've kind of gone from a tree of life and everything down to cetaceans, down to delphinids and killer whales and ecotypes and the resident ecotype. We can go even further and talk about acoustic clans. Those are within in populations, and those are groups of killer whales that have kind of the same dialect or the same sounds. And then pods, which are kind of groups of related killer whales. So for the southern resident population, we have JK and L pod. And then macrolines, which I'll be kind of focusing on for the rest of the talk, which are females and a female and her immediate descendants. So as Susan mentioned, the southern resident killer whales were listed as endangered under the Endangered Species Act in 2005. The main risk factors that cause them to be at risk are, one, simply a lack of population growth and a small population size. So simply their demography is such that they are at risk from just small population size effects. And then potentially a lack of prey quantity and quality, contaminants, and then disturbance from boats and noise. So our research program at the Northwest Fisheries Science Center focuses on all of these. Today I'm really only going to be talking about the breeding structure of the population, and that's relevant to this risk associated with abundance and demography. So how does breeding structure affect risk? There's a couple of ways. One, genetic diversity, as we sort of learned from Darwin, is important for allowing populations to adapt and respond to environmental changes. Diverse populations have what they need to do that. And the breeding structure affects how much diversity there is in a population. Both how much, if you have interbreeding with other populations, that can increase diversity. If you don't, it reduces diversity. And then even if you don't have interbreeding with other populations, kind of how the breeding structure occurs within a population affects how much diversity is in that population. So for example, the number of breeders, if you have most of the population breeding with kind of an even variance in breeding success, that tends to promote genetic diversity. If you have only a couple breeders, that tends to reduce diversity. And then whether relatives interbreed. So if you have inbreeding, inbreeding tends to reduce genetic diversity compared to non-inbreeding populations. And then finally, the breeding structure, some aspects of the breeding structure can simply affect actually maybe even just the population growth. So for example, if there are certain customs or taboos or something that limit breeding, say on pods or something, that could potentially actually limit population growth due to a lack of opportunity for appropriate mates. So all these things are kind of hypothetical in ways that the breeding structure might affect extinction risk. So why do we use genetics to study this? And there's a couple of reasons. One is that there's often a really big difference between the kind of social breeding structure that you might observe simply by watching a population and what you learn when you actually look at the genes or see kind of really who's breeding with whom. And a great example of this is with birds. So in 1970, the kind of state-of-the-art general feeling amongst people studying passing birds was that they were nearly all monogamous. And that's because when you go out and look with a pair of binoculars and kind of watch who's mating with whom and who's nesting with whom, it sure looks like they're monogamous. But over this time period when DNA sequencing became available and scientists could actually study the transmission of genes in these birds and figure out who the dads were, it turns out that actually they're not monogamous at all. And most passerine birds are in fact polygamous. And the kind of social dad who's on the nest is not the only dad of the chicks in that nest. And the same is true for marine mammals. So, for example, in seals, you can go out with binoculars and kind of watch who's moving with whom, and you might kind of get the idea that the big dominant male is the only guy doing the breeding. But it turns out when you go into the genetics, a lot of the pups are sired by more kind of junior males, if you will, and those matings are occurring in the water where you can't see them. And so you wouldn't know that if you just watch the population and do the genetics. And then finally, a lot of things like whales or other animals that are difficult to study, it's usually easier to get a genetic sample than it is to observe the animal in some cases. So if they're in an aquatic habitat, for example, or if they're simply not observable for much of the time, it might be easier to do this with genetics than observation. So for studying killer whale opportunity, the first thing we have to do, well actually, let me back up slightly. In the case of killer whales, the maternal relationships, so basically who the moms are, is well known through decades of study. So starting in the early 1970s, Canadian researchers like Michael Bigg, and then later John Ford and colleagues would go out and watch the whales, and by observing the whales, you can pretty much, and then up here in this area, Ken Galcom and Center for Whale Research started this research in the mid-70s. And simply by observing the whales, it's fairly readily apparent who the moms are. A calf shows up, she's with her mom, he's with her mom, and so the maternal relationships and who the moms are relatively well documented. The question is who are the dads, and that you really can't figure out by watching the whales. So to figure that out, the first thing you have to do is start to get tissue samples or some way to collect DNA from the animals in the population. And we do this in two different ways. The first and kind of most effective and important way is to collect a biopsy sample. So here's Brad Hansen, who's the head of the marine mammal team at the Northwest Fisheries Science Center. And he's got a pneumatic dart gun here and is collecting about an inch long sample from a whale, about the diameter of a pencil. And this is our most important source of DNA from these whales. And the reason why this is a great source of DNA is twofold. One, because you're getting a lot of, a little bit of the skin, you get a very high quality sample, so the DNA is easy to use and easy to study, and you get a fair bit of it. And then two, kind of most importantly, because you're right next to the whale, when you take that sample, you know exactly what it is you're sampling. There's no ambiguity that this sample came from this particular whale. Our other source is also an important source, but it's maybe slightly more difficult to use, and that's fecal samples. So there's a lot of DNA in poop, and we use these samples for all kinds of things, particularly for studying the diet of the whales, where it's invaluable. But we can also get whale DNA out of the poop samples. The sort of cells from the intestinal tract end up in the poop, and you can get whale DNA from those. So these are collected basically by following the whales around in a small boat with a swimming pool net and simply scooping the poop. These are great samples. We collect a lot of them this way. We also collaborate with Sam Wasser at the University of Washington, and he and his students use dogs who have been trained to smell out whale poop and collect it on the water. So we get a lot of samples like this, many more poop samples than biopsy samples. The one kind of weakness of these samples is that we typically don't know exactly who did the pooping, particularly with the dog samples, which are not collected near the whales. So these are kind of an important secondary source of samples. At this point, we actually have most of the population biopsy sample. So how do you do a paternity analysis? It's really just like, it's kind of using the same principles that Mendel used and discovered in his peas. So, as I mentioned, there's about 3 billion base pairs in the genetic code of a killer whale. We go in and look at, for this particular study, we're looking at about 90 of those 3 billion sites, but that's enough information for us to figure out who the dads are. And so the principle of that is illustrated here. So we have a mom killer whale, and we have a group of several dads who are the possible dads of this mom's 2 calves. And so in this case, we go in and we sequence the mom, and at this particular place in these 3 billion base pairs, we discover that this mom received a T allele from her dad, and she also received a T allele from her mom. So she's a TT. We go in and look at her calves, and one of her calves is a CT, and one of her calves is an AT. So we know that T had to have come from mom, because she's just a TT. So in this case, the A and the C of these 2 different calves had to have come from the dad. With me so far? So we then go and look at these dads. So we got one dad who's a TT, another one who's an AA, and another one who's a CA. So from this, if we're confident that these 3 are the only dads who are the only possible dads, and if we kind of rule out things like mutation or other things that might screw things up, we can say with some confidence that the C dad has to be the father of the calf that has the C allele. With the other calf, it's a little more complicated because we have 2 dads who have A's. We have a CA and an AA. And so either one of those dads could potentially be the father of this calf over here and contributed that A allele. And, we might count things up and say that, well, the dad who has 2 A's is maybe more likely to be the father than the dad who just has 1 A's, and that's true. And so we might say he's a little more likely, but we can't rule it out entirely. So this is the principle we use for paternity analysis. Instead of looking at just one site, we look at a whole bunch, which gives us a lot more power to do that. But that's the basic idea. So let's look at a real world example of this. So this is female K14 on the left and her calf K42 on the right. K14 is one of the older females, born in the mid-70s, and her calf was born in 2008. And the question is, who is dad? And drum roll. There we go. So here are all of the potential dads. So because this one was born in 2008, relatively recently, we actually have samples from, there's basically 20 dads, roughly about 20 dads in the population, 20 males, sorry, in the population who could be this calf's dad. And here they are. And we have samples from basically all but one of them. There's one dad who's not here, but we have samples from everybody else. Nearly all the dads are genetically sampled. And you can see, the way we do this is to kind of give every male the benefit of the doubt. So we assume that any male over the age of around 10 or 11 is at least a potential dad. And so some of these look pretty different. So for example, this picture here, we have L41, and then L88 and L89. L41 is an older male, very mature. These were not, these 2 at the time of this picture. Sorry, I should not use the laser pointer if I accidentally move things. These files are big enough that you need to go slow on hitting the buttons here. Okay, here we go. Anyway, so in that bottom picture, you have like L41, who's a very large, very mature male, and some younger males who are included in the group of potential males, but are less mature and probably less likely to be the dads, so we give them the benefit of the doubt. So in this case, we go through our analysis, and it turns out it is. In this case, L41 was the dad of K42, and there's no ambiguity in this particular result. There's really no other dad that could be. Turns out he's also the father of K42's older brother, K36. So those two share the same dad, are full siblings. But he's not the dad of their other sibling, K26, who has some other dad. And in fact, K26's dad is not among the ones that we have sampled, so he remains an unknown father. Probably because K26 is an older whale, and his father presumably died before we had an opportunity to sample. In some cases, so for most of the assignments that we do, we know who the mom is, and we're just trying to figure out the dad. But we sometimes end up in situations where we don't know who the mom is. These are often kind of sad situations, but important nonetheless. So amongst our samples, we have 3 or 4 cases where we have a neonate calf or some young, very young animal who is washed up on the beach somewhere before it was ever observed in the population. So you don't know who the mom is, and we're interested in knowing both who the mom and the dad is. So here, for example, this calf, this neonate calf, washed up on Benjamin Spit in 2013, and we had the opportunity to try to figure out who the parents were. In this case, we could show that it was the mom was J28, and the dad was also L41. So, what does this look like if you kind of put it all together? And this is where you might want to use civil trial. It's a little hard to see. That's okay, because let's deal with the colors here. So this is a family tree of the whole population that shows every individual in the population who has ever been observed is a dot on this tree. So each of these little dots that you see are an individual whale, some of whom are dead, but it kind of shows the entire population. And they're ordered from bottom to top in their birthdays. So the older whales are at the bottom, the younger whales are at the top, going from the early 1900s up to the present. These older dates are kind of guesses. Each mom and calf is connected by a line, which I acknowledge is a little bit hard to see on the screen. And so all the match lines you can see are kind of along the screen here. The dads are color-coded, so each of the dads is a triangle. So here's one dad, this is J1. Here's another dad, this is L41. And there's a total of 8 males that have been identified as fathers in the population. So J1, J26, K26, L41, L57, L78, L79, and L85. There's a couple things to kind of notice here. One is that, then each dad is connected to his offspring by the color. So everything that's red in here are offspring of J1. Everything that's blue, those are the 2 dominant ones, are the offspring of L41. And the others are color-coded accordingly. So there's a couple things to notice. One, we have a lot more paternities in the more recent data, and that's simply because we have those fathers sampled. So we have no idea who the dads are of these older animals who were born a long time ago, because their dads have been lost to history and never were part of our sample. So most of our information is based on younger animals. And then the second kind of obvious thing from this is that the variance in breeding success is pretty extreme. There's basically two animals here who are responsible for 85% of the paternity assignments, and those are J1 and L41. So what do we know about these guys? So you have J1 on the top, L41 on the bottom. J1 has been the oldest male in the population, was the oldest male in the population throughout the entire study period. He was he's one of the larger males, but not the largest. And then L41 is now the oldest male in the population since J1 died, and he's also the largest male. So basically, the oldest and largest two males are responsible for the vast bulk of the offspring production. You can kind of look at this statistically. This is a graph looking at the probability that a whale is a dad versus their age. And you kind of see that basically over time, the older you are, the more likely you are as a killer whale, in this population anyway, to be a father. Our youngest paternity assignment so far is at about age 16, and the oldest is at age 59. So I want to talk a little bit about inbreeding. One of the things that we're interested in doing this study is to kind of evaluate whether there is a problem with inbreeding in the population and whether it could be one of the factors leading to the population's status. So inbreeding is simply mating between relatives. It leads to lower genetic diversity, and if you think back to Mendel's P's, the sort of gene for shortness, that gene for shortness only manifests itself when you have two short genes coming together in the same individual. When you have matings between relatives, because they share a common ancestry, they're more likely to be genetically similar, and you have a higher probability of breeding two of those same types of genes together in the same individual. Inbreeding depression is the reduction in fitness, either survivorship or say calving rate, it's caused by inbreeding or mating between relatives. And it's primarily due to the fact that you're bringing these genes together in the same individual. So all of us in this room, and most mammals, most organisms have a large number of deleterious genes in our genome. And for the most part, that doesn't harm us too much because they're paired with a good gene. Basically, you might have gotten a bad gene from mom, but it's okay because you get a good gene from dad or vice versa. When you have inbreeding going on, you have a higher probability of bringing two of those bad genes together at the same time, which can lead to reduced fitness. So the question is this a problem for the population? It can definitely be a problem for some populations. So here's an example of a recent study in red deer in an island in Scotland that shares a lot of similarities to the southern resident population. These deer also form natural lines. They're about the same size of population, about 80 to 100 individuals. And remarkably, they've actually been studied over exactly the same time period. This population was first observed and subject to study in the mid-1970s and has been followed ever since in a manner very, very similar to the southern resident population. And here's just sort of an example of inbreeding in this population. All these graphs over here just show a relationship between inbreeding on the horizontal axis, where you're going from low inbreeding to high inbreeding, and then different measures of fitness. So, for example, these two lower graphs here are lifetime breeding success for females and males. And basically, as you become more inbred, particularly for males, but also for females, your breeding success goes way down in this population. So there's sort of an example of what inbreeding depression is. So are there any reasons to be concerned about this? In the southern resident population. And we don't really know the answer yet, but there are some reasons, I think, to be concerned. One is just as you kind of see from these colors up here, at this point, roughly half of the population actually is descended from either J1 or L41. So those two males, if you kind of trace back of all the roughly 80 individuals who are currently in this population, about 35 of them have either J1 as a father, grandfather, or great-grandfather, or L41 in that category. And actually 5 of them managed to have both J1 and L41 as an ancestor. So clearly, as you kind of think towards the future here, it's going to be hard to avoid some degree of inbreeding because a big portion of the population is basically half siblings, and there's really no way that they can avoid mating with each other. So kind of thinking in the future, you're probably going to have some of that happening. Whether we can detect this right now, whether we can detect inbreeding depression right now, is difficult to say, though. In order to kind of evaluate inbreeding depression from a pedigree like this, you really need 4 or 5 generations to calculate an inbreeding coefficient effectively from a pedigree. And for most individuals, we don't have that. For some individuals, we do, but you need to have a lot of individuals in that category. For the most part, this pedigree is not that deep. So we take kind of a shortcut to looking for inbreeding depression here. And that is, as you recall, I said that inbreeding leads to lower genetic diversity, and to some degree it's that lower genetic diversity that causes inbreeding depression. So we can't look at this directly and just say, is there any relationship between a whale's genetic diversity and its fitness? And so that's what we're doing here. So this is a graph based on the data that I'm just showing you of the diversity in a whale's genome on the horizontal axis, going from high diversity over here to low diversity over here, and the probability of annual survival from year to year. And this is a fitted model here, where the line is the kind of best fitted model, and then this band kind of shows the variance around that fit. And so you can see that maybe there's some evidence here that we have inbreeding depression going on in the population. But those individuals that are less diverse have a somewhat lower probability of survival. But there's a lot of uncertainty about that relationship at this time. So one of the things that we're going to be working on over the next year or so is trying to tighten that relationship by collecting more genetic data to better answer this question. There are a few other, you can't really see it in this picture, but there are a few other causes for concern. Unlike the last time in 2011 we published a paper looking at our initial results of the pedigree, and we didn't find any evidence of close inbreeding in the population or inbreeding between really close relatives. That's changed a little bit now. We have a couple instances in this pedigree where we have evidence of fairly close relatives producing offspring. So there's a little more evidence of actual inbreeding going on in the population now. So this is basically my last slide, and I just kind of want to leave you with a question or idea of whether this might be a factor that explains differences in the population status between different killer whale populations. This is a figure from a recent paper by Eric Ward at the Northwest Fisheries Science Center and Marilyn Dahlheim at the Alaska Fisheries Science Center and their colleagues. And this is looking at on the top, this is the birth rate and survival rate of the southern resident population in the continuous line and the Southeast Alaska resident population in the dotted line over time, from the mid-1980s to the present. And what you kind of see in this is that basically over time, the Southeast Alaska population has had a consistently higher birth rate over this time period. And for the most part, it's a little more subtle, but also a consistently higher survival rate, particularly during this time period in the mid-1990s. And because of that, the population of Southeast Alaska has more or less tripled or almost quadrupled in size over the same time period that the southern resident population has really not grown at all. And there's a lot of reasons why this might be the case. It's Alaska, it's more pristine, maybe there's more food, less contaminants, and so forth. But I think we can maybe also add the potential that maybe the southern resident population is also more inbred as a potential alternative hypothesis to look at in understanding these differences among these populations. So with that, I will reach the end, and thank you for your attention, and I'd be happy to address any questions that you might have.
population would have been of the southern residents pre-European arrival. And obviously the carrying capacity would have been much bigger just because there would be more fish. But there's probably other factors too that limits. You have what's your feeling on that?
Yeah, so that's a hard question to answer, unfortunately. I think it's pretty, my understanding is, if you just sort of count up whales that were taken, into the aquarium, trade or killed or captured in the 60s and 70s and kind of add it to the population size at that time, you get up to, I think, about 120 some odd whales. So you kind of, that level is clearly documented. And if you look at, things like the northern resident population, which is now, with roughly 300 whales or so, it's easy to imagine that there could have been a couple hundred, I think. And certainly, the Columbia River, prior to the early 1900s, was the center of distribution and abundance for Chinook salmon, which is the preferred prey of this population. And so it's easy to imagine that it could have supported a pretty large group. Now that being said, those fish mostly swim north and also support a lot of killer whale. The southern residents and the populations further to the north do to some degree share the same prey base because of the distribution of the salmon. And so the Columbia was not just supporting the southern residents, it also is now also supporting the more northerly populations. There's some hints in the genetic data. There are methods that you go in and kind of ask, are there signs that this population used to be bigger? And there are some hints in the data that it did used to be bigger, but you can't really use that to say how much bigger, unfortunately, at this time. One of the other things from the pedigree that I haven't really explored yet, but I'm interested in looking into is, if you look at the current pedigree, and again, this is hard to say here, but there are a lot of full siblings amongst the population right now. So there are many cases where a female and her offspring, where the offspring of the female is produced have the same father. And you see that in certain, especially in J-pod. And even though these older animals, we don't know who their fathers are, you can't ask what their sibling relationships are. And there are no full siblings in the older animals. None of the older animals that I've sampled are full siblings. A couple of them might be half siblings, but they're full siblings. And that, I kind of want to think about that a little bit, but to me it kind of suggests that maybe the population was bigger in the past, or at least the breeding structure might have been a little different. Because you don't see these older animals.
But I mean, it couldn't. Do you think it could have been like 1,000 individuals, or I mean, how
well,
big?
I don't think so myself. You don't really see that in these resident populations, so I don't think they get that big. Just naturally, I think they split. But sorry, yes, go ahead.
I'm wondering, with the inbreeding depression, if it impacts the sex of the youth that are born.
Yeah, I don't know. It's a good question. I think, there's been some concern over the kind of sex ratio of recent births and kind of the sort of dominance of males, which is concerning for future population growth. We really need females to get the population to grow. We did look at that, actually, tried to find out, ask if there was any tendency, for example, of offspring of J1 or L41 to, be, one way or the other. And there's no statistical evidence of that at this point. And in fact, there's really not much statistical evidence for deviation from 50-50. It could just, at this point, be bad luck still that there's so many males that come.
Yes, in your survival rate slide, it showed in the mid-90s a sharp decline for the southern residents, while the Alaska residents maintained fairly constant. But then at the most recent years, it shows both of them plummeting. Is there correlation there, or why is it different now than it was in the mid-90s?
Yeah, it's really interesting. I don't know. Particularly if you look at those paternity rates at the top, they are highly correlated. So even though the Alaska population is doing a lot better, it goes up and down in the same pattern that the southern residents do. And the survival rates are pretty correlated too, with the exception of that period in the mid-90s. And the inference from this paper, you know, that certainly the sort of answer to that these authors gave is that they share the same prey base. They're probably being driven by the ups and downs of the salmon runs. And this, you know, this period of the mid-90s was a period of particularly poor salmon returns. So yeah, I don't have a good explanation for why they deviated so much in the 90s. I think that in general, these more northern populations might kind of have first access to the salmon, if you will. You know, these salmon tend to migrate, most salmon tend to migrate north. It may simply be that they have a better opportunity in low years to access those salmon than the more southerly populations do. Sure, in the back right there.
Do you see this research leading toward any new knowledge that will help us save the southern residents?
I think that one of the premises of kind of the whole research program is that more information on what is causing the decline is going to be helpful in finding solutions to that. A lot of that involves looking at correlations between the population's birth or death or whatever, and different factors, whether it be salmon or boat traffic or so on and so forth. So I think the more information we have on kind of what factors actually do influence individual level variance in fitness, that kind of allows us to better understand, factors that we might be able to do something about. I don't think we're going to directly interfere with the population to influence its mating patterns or anything like that. I'm not going to do that. But I think knowing, if in fact, say the degree of individual genetic diversity is an important predictor of whether a whale is going to have a high fecundity rate or a low fecundity rate, that's important information for, then asking, understanding other factors, such as, say, prey abundance, that we might be able to do something about. Yeah.
Is there any evidence that the resident orcas are seeking other food sources than salmon, with the reduction of salmon?
Well, they definitely eat other things than salmon. So we do a lot of work on the diet, and I mentioned those fecal samples. Those are particularly useful for studying the diet. We basically just go in and sequence the DNA that we extract from those fecal samples and ask what species that DNA belongs to. And from that, they clearly eat a lot of Chinook salmon, and when Chinook salmon are available, they seem to prefer, they choose them and they eat them. But they eat a lot of other things too. So in the late summer, they switch over to high fractions coho salmon, up to 50% coho salmon. In the fall, they switch to up to at times 100% chum salmon. The data that we have for the winter shows an even more diverse diet. It's still kind of dominated by Chinook salmon. But there's a bunch of non-salmonids. They eat some halibut, lingcod, some skates and rays, and stuff. So it isn't just salmon. They're clearly capable and do eat other fish.
Have you detected any evidence or any sense of the impact of climate change on the well-being of the orca?
Not directly.
Can you repeat the question?
Oh, sorry. The question, my apologies. The question was, do we have any detected any evidence that climate change has affected the well-being of the orcas? No, I'd say not so far. I mean, you can kind of guess to the degree that climate change influences their prey base. I think that would be the mechanism that orca themselves will be directly impacted by changes in ocean temperature, that sort of thing. I think they're sort of flexible enough that they can handle that. But degree that climate change impacts salmon abundance or other prey abundance, that would impact them. And so far, I think the jury's kind of out. You know, the work we've done on climate change and salmon, you know, suggests that particularly in the interior of Colombia and other areas where temperature can be a limiting factor for salmon, climate change is likely to be pretty bad. But there's a lot of uncertainty about kind of what's going to happen with these salmon stocks. So the short answer is no, we don't think about it. Sure. More. I'm sorry.
We only have time for.
Oh, sorry, Mike. I don't want to take point on that. One more question, I guess.
Yeah, we could go on.
You mentioned earlier that might be some cultural taboos per se for breeding, but in 2015 it was that 2 fairly old females had calves in their 40s, and then at least 2 of the very young 10-year-olds gave birth. Is that something that has been seen in more healthy populations, or is it kind of an anomaly?
No, I don't think it is an anomaly. And I sort of mentioned that because some of the earlier work on both the southern resident population and the northern resident population suggested, for example, that there wouldn't be much interbreeding within a pod. So that's kind of what I meant by a social custom or something. And you know, so far we're not finding anything like that. They're pretty much just randomly, you know, there's definitely interbreeding within a pod. And we don't really detect any